Hi everyone, welcome to today's seminar. It's a great pleasure for me to introduce Jorgo, who's going to be our first speaker from the university wing to give one of these seminars. So he started his scientific career here in Cambridge studying natural sciences. He then went to do a PhD in Heidelberg in Germany, and then to the US, where he did postdoctoral work at Harvard Medical School. He then became an associate professor at Yale University and then did full circle and came back to the University of Cambridge, where he's been since 2014. And today he's going to tell us about his favourite subject, which is viruses, what they look like, how they spread, and why you need them. So thank you.
Great. Thanks. Thank you. Thanks for the introduction and for having me here today. It's a pleasure to be here and share our work and insights with the non-scientific staff of the LMB. I'll just start by introducing my group. We are in the uni wing, so this is the southwest wing on the first floor. You may recognize some of the faces. This is Emma, this is our lab manager for the whole wing. And yeah, as Alison said, we're affiliated with the University of Cambridge Department of Medicine. So there's us and 3 other groups there. And hopefully today we'll give you an idea of the kind of work that's going on in that part of the building. So so we work on viruses. That is our favorite subject. And so why study viruses? Well, the obvious thing is that they're the cause of many important and medically important infectious diseases. So viruses cause all sorts of diseases and problems. And so our first goal is to know thy enemy. In the words of Sun Tzu in The Art of War, know the enemy and know yourself. And that's really where we are. We're trying to understand in detail how viruses work, what they look like, and how our bodies respond to viral infection. And so our strategy to know our enemy is to obtain really detailed pictures of what viruses look like on the molecular and atomic level, because this allows us to recognize what tricks they're using and explain how they work and how they get into the cell and how they infect your body and perhaps how we might get in the way and stop some of those processes. So ultimately our goal is to help fight the spread of disease, of viral disease. And although we're based, really very much at understanding the general principles, we always have in mind, how are we going to develop a new antiviral strategy or a new vaccine, and understanding each step of infection, each step of how a virus progresses its way through the different stages of infection, starting with entering the very first cell in your body, will help us identify some of the vulnerabilities, some of the weak points, some of the opportunities where we can intervene with a drug or a vaccine to stop the infection. And so that's the undercurrent in what we do. And what I'll tell you today in the second part of my talk is about a specific stage of infection, the very first stage, where a virus is coming in and it's trying to force its way into the cell and it's trying to deliver its genetic payload, its genome, its DNA into your cell. And I'll talk about a very specific step called the fusion step, where the virus merges with the cell and delivers its genome into the cell, and how we might possibly be able to interfere with that or stop that in its tracks. And then in the third part of the talk, I'm going to tell you about actually why you might need viruses and how we've come to perhaps rely on viruses and us as a species and actually all species. It turns out viruses have been around essentially since the origins of life, as far as we can tell. They've been around, since the very beginning. Bacteria are infected by viruses, and some of these viruses are harmless, and actually some of them turn out to be useful, and we've co-evolved to become dependent on them. And I'll show you a couple of examples of that at the end. So first I'm going to tell you about obtaining the detailed pictures of viruses, right? So this is to really understand what they look like, how they work. And so our favorite approaches here are using these electron microscopes, which many of you probably aware of. There's 3 of these in the building. Each one of these is 4 or 5, 6 million pounds, depending on how you kit it out. And there's a few here. There's also some that we use at the European Bioimaging Center near Oxford. And this is Chin Yu from the lab, who's become a real expert in using these microscopes. And these allow us to get directly image these viruses and get really detailed pictures. And then sometimes that's still not enough, and we want an even more detailed picture of some of the building blocks of the viruses. And in that case, we use this facility here. It's a European synchrotron research facility in Grenoble, France. And what it is it's a super bright X-ray source. And so the X-ray, you have electrons, or you have particles spinning around here and producing really, really bright X-rays. And that allows you to image the building blocks of viruses and any other living thing at a really, really high resolution, really, really detailed, with a lot of rich detail. So those are the approaches that we're using. And then the kinds of images that we get are hard to represent because they're on such a small scale, and I'm going to come back to that. But this is more or less what a typical spherical virus looks like. You may recognize the symmetry here because it's the same symmetry as a football, right? So it's called icosahedral symmetry because it has 20 repeating units. And you can see the soccer ball has these pentagons, and in the virus there's 5 pointed stars in this virus and in other ones. And actually they share the same kind of overall assembly and symmetry as a football. And it's a recurring theme among viruses. Not all viruses have this, but a large fraction of them do. And the reason they adopt this type of assembly is that it's a very rigid and sturdy and strong assembly, and it allows the viruses to withstand the environment and the different sort of challenges that they might encounter when they're outside of the body waiting to infect somebody. And so this is the Epcot Center in Florida, and it has the same kind of thing, and it's assembly. It's an icosahedron, and it's just a very sturdy, strong assembly. So this particular example is dengue virus, and actually Zika virus looks the exact same at this level of representation. They're very difficult to distinguish at this level of detail, which is why I said sometimes that's not enough. We need even more detail. But other viruses have different types of assemblies, and they come in all shapes and sizes. But all of these viruses here, all these virus particles I'm showing here have the same icosahedral symmetry. So the details of the assembly might be different. Some of them might be a little bit less spherical than others. And then there's a big difference, a big range in sizes. But they all have this same kind of symmetry. And each one of these is usually an electron microscopy structure, or it can be based on the X-ray structures that I showed you. And it gives us a really detailed picture of what these look like. So the virus I showed you is in the middle here, and there's much smaller ones and much bigger ones. And this is actually a scale bar. So this is giving you the scale. And I realize many of you might not know what a nanometer is, so I want to spend just maybe 2 minutes kind of really giving you a sense of how small these things really are. So if this is a virus, this is 100 nanometers, this scale, this bar, this double arrow here. 100 nanometers is 1/10,000th of 1 centimeter. So that's how small it is. And to give you an idea, if a virus was the size of a football, a typical football, 20 centimeters, then a human body would be 200 kilometers from head to toe, right? So that's kind of the comparison. And here I have an animation that really is going to zoom in from the human scale down to the nanometer scale to really sort of drive home the point of what scale we're talking about here. So we're going to zoom in, and we're at 1 centimeter here. We're going to zoom all the way in until you see cells, and here's some blood cells and white cells. These are your white blood cells and your red blood cells. And then inside the cell there's DNA, which we're going to talk about later. And then inside the DNA, it's made up of these little double helices that we have one at the front of the building. And if you keep zooming in and stop right about here, that's 1 nanometer. At 1 nanometer, you can see individual atoms. And so really it allows you to it gives you atomic resolution in your picture. So we've done this kind of imaging with viruses where we actually know we can model where every single atom in the virus is. And so if this is an electron microscopy reconstruction, if we zoom in into a crystal structure or a really, really good electron microscopy structure, we can see where the individual atoms are, and these little sticks are representing the positions of individual atoms. And why this is useful is because now we know exactly what the virus looks like, and this is what our body sees. This is what our immune system sees when a virus comes in. It's the outside of the virus, and we know every shape of and every nook and cranny on the viral surface. And now we can start thinking about ways to maybe cross-link these or lock them together or prevent the virus from attaching to a cell, all sorts of strategies like this to stop the virus in its tracks. And so the one step that we're particularly interested in is the virus entry step, how the virus first gets into a cell, how it forces its way into the cell. And so many viruses have a flexible envelope under the protein shell. It's called a lipid membrane, but it's really just a flexible envelope. And it's the exact same kind of envelope that each one of our cells in our body has, sort of keeping the contents in and preventing them from spilling out. And these membranes, are protective because they prevent things from diffusing in and out, and they keep the cell secure from the outside, and particularly from viruses. But viruses have ways of getting in, and so usually what happens is that they come in into a little compartment, into a little bubble inside the cell. So this is a cell, this is a virus, and it's made its way into one of these so-called vesicles, into these compartments. But what it still needs to do is get its genome, its DNA, into the cell so it can be replicated. And the way this works is that the viral envelope has to fuse with the cell envelope. And so going from this panel here to this one, there's a fusion event that involves the merging of the viral envelope with the cell envelope so that you end up with a single continuous envelope, a single membrane. And there's a protein, there's a bit of machinery on the outside of the virus that actually drives that process, that drives the fusion of the two envelopes. And we've actually learned a lot about how this works in detail from our imaging studies and getting the detailed pictures that I've just been showing you. So this is the envelope protein, the building block of the virus that I showed earlier. That's what makes up this outer shell that I was showing. And this is how it exists in the mature virus. But actually, when the virus starts fusing these membranes together and undergoing driving this fusion step, it changes shape. The change in shape is what actually drives the membranes together. What we were able to do is to determine the structure in detail of what the envelope protein looks like after fusion. So these are two snapshots of the same bit of machinery on the virus, the fusion machinery, before the fusion event and after the fusion event that delivers the viral DNA into the inside of the cell. And so by looking at these two, we can start piecing together the different steps that allow the virus to enter a cell, that allow the virus to deliver its DNA into the cell where it can get replicated. And so this is our cartoon that we've drawn up based on our observations, our detailed structures of snapshots before and after. And you may recognize this is the pre-fusion structure here, there's the post-fusion structure here in kind of cartoon form. We can start drawing what the intermediate steps might look like. And what we're doing here is we're building a molecular movie, if you wish, of what the different steps might look like of a virus actually entering a cell. And this is still pretty abstract. We've got the virus protein opening up and then dipping in. This is the cellular membrane, and then the two membranes kind of get pulled together, and there's these contortions that finally allow the two membranes to fuse. But it's still a little bit difficult, I think, to picture this. So I have another movie that's going to show you, I think, in more 3D, a little bit more intuitively, how we think this is working. So this is a virus inside of one of the compartments I was telling you about. And what happens is that it kind of opens up. You can see the outer shell, the protein, this is the envelope protein, opening up and arranging itself so it's poised to insert itself into the cell membrane. And this is the cell membrane. This is the membrane of that compartment. And these two envelopes now, the viral envelope and the cell envelope, are going to be brought together by these two envelope proteins on the viral surface through this series of changes in shape and contortions, conformational changes we call them, that allow the two membranes to be kind of pinched together so that they merge. And when they merge, that actually allows the DNA that's trapped inside the virus to pour out of this, through the inside of this stalk into the inside of the cell. We're now viewing it again from the other side. So this I think really gives you a step-by-step view of what's happening on a molecular level on a scale that's really too small to see with light when we have to use electron microscopes to visualize. And this isn't, I mean, it's a nice cartoon and everything, but hopefully it actually has some real use. And in this case, one potential use is that we can think about blocking some of these steps. And so very specifically in this case, remember how this little bit here is going to anneal here, and these two membranes are going to fuse together, and I said that was a key step in entry. Well, if you design a drug that could bind really tightly, that could stick really tightly to the surface, it might prevent this component from binding, and it might just stop that process in its tracks. And so that's actually an antiviral treatment strategy. And there's a clinically approved drug against HIV that works in this manner. This isn't HIV, and we're hoping that this might also be applicable to some of the other viruses that we're studying. All right, so in the last few minutes, I'm just going to move on to the last bit, which is why viruses might be useful and what's going on there. So it turns out that there's a whole set of viruses that can, including HIV and retroviruses in general, that they can take their DNA and insert it into the human genome, into your genome. So when this type of virus comes in and delivers its DNA into the cell, like we've just saw, that DNA can be just inserted into your DNA. And what that means is that every one of us slowly acquires viral DNA. And this has been happening for a long time. And when viruses infect an egg cell, or a sperm or an egg cell, that viral DNA can be transmitted to future generations. And it happens fairly rarely, but over the thousands and millions of years that we've been evolving, it's happened many, many, many thousands of times. And so our human DNA actually has a lot of viral DNA that we've inherited from ancestral infections, our ancestors being infected by retroviruses. So actually 14% of our genome is endogenous. We call them endogenous viruses because there are sequences that have become part of the human genome from previous infections. And another 34% of our genome is virus-like elements. That it's not quite a virus, but it has virus-like properties. And if you compare that, only 2% of our genome is making our proteins, is making the proteins that make a human body. And this whopping 48% is virus-like elements. And another 50% of our genome is actually regulating how much of the proteins that we make and actually has unknown functions in a large part. So there's a lot of the human genome, even though we know all the sequence of it, we really don't know a lot of the function of it. And there's this huge fraction that comes from viruses. And you might ask, why have we retained these elements over the millions of years that we've been evolving? And why do all species, not just humans, have a large amount of viral DNA left over in the genome? And for a long time, we thought this was just junk, that it was just kind of a evolutionary relic or a fossil, if you will. But actually, it turns out that there's some important functions in these genes. So I'll just give you 3 quick examples of functions where the viral DNA has become important. And one is in making the human placenta. So, the human placenta is what separates the fetus from the mother, and it's very important in keeping the bloodstream of the mother and of the fetus separate. And what keeps those two bloodstreams separate are this dark layer here, which actually, if you zoom in, is made up of fused cells. And the cells have undergone fusion, and when an embryo first implants, this is a really early human embryo, it's surrounded by a layer of fused cells. And it turns out that these cells are fused by a protein, and it's an envelope fusion event that fuses the cells that looks a lot like the fusion event that is required to fuse a virus to a cell that I was showing you earlier. And it turns out that if you look around the virus species, there are retroviruses like this one, HTLV1, that have fusion proteins that look just like the fusion protein in humans that makes placentas. And so what we conclude is that actually what's happened here is that a viral sequence that became part of our genome at some point, and specifically when mammals diverged from other species, that sequence was taken out and repurposed by the human body, by mammals, to make placental cells. And so what we think is that these viral sequences are kind of a genetic toolkit where you have all these sequences that aren't actually being used actively, but now and then something becomes useful and it gets grabbed and it gets taken up. One other function, just briefly, is fertilization. So again, fertilization, what it is it's the sperm fusing with the egg. It's a cell-cell fusion event, and it happens in plants, it happens in animal cells, and in every case you need the sperm cell to fuse with an egg cell. And we know in the case of plants that the protein that does that is this protein called HAP2, and this is what it looks like in detail. And if you compare it to the viral proteins, there are viruses that have proteins that are also fusion proteins. It's a different kind than I showed in the last slide, that look exactly the same or very similar, recognizably similar. And so what this is telling us is that at some point, either a cell borrowed from a virus or a virus borrowed from a cell, but in any case, viruses and cells share this machinery, this fusion machinery, that in some case, that in animals and plants is being used in fertilization, and viruses are using it to fuse themselves to their host cells. So this, I think, gives you an idea, and this is the last slide, that viruses and cells are closely intertwined evolutionarily, and that we've been evolving so long side by side that actually we share common genes and machinery, and we have viral DNA within us that is often has been repurposed over the millennia, and that has grown to fulfill really key functions in human biology, including fertilization and placenta formation, for example. But we actually think there are many other examples that still remain to be discovered. So with that, I think I'll wrap up. I think I'm out of time. This is my group. I'd just like to thank everyone in the group. But for today, I'd really like to extend a big thank you to all of the support staff, non-research staff, administrative staff, operations that really keep the Institute running and make this a great place to work. So thank you for your attention. I'll take any questions.